So if you remember, we saw earlier that around about the middle of last century, the hippocampus was starting to emerge as a potential memory structure, and that's because of the case of Henry Mollison, who had had a bilateral temporal lobectomy to try and cure his epilepsy, and he woke up from that surgery with this very dense amnesia, and that suggested to Scoville and Milner that the hippocampus might be a memory structure of some sort. So the hippocampus became the focus of a lot of neurobiological interest from that point onwards. This is just to remind you that the hippocampus is a very intricate and beautiful structure, and you can see that its cells are arranged in these very regular layers, and something about the regularity of their organization suggested that the hippocampus is doing some common operation on all of the varied information coming through, and that operation might have something to do with memory. So to get at that question, it becomes necessary to actually investigate the hippocampus electrophysiologically. And we've already encountered that with studies of LTP and these studies of evoked potentials when you're recording lots and lots of neurons all being stimulated simultaneously with electrical impulses. But really, to look at memory formation spontaneously, we want to be able to look at individual neurons. And that's a much more difficult task because an individual neuron is very tiny. It's only 50 microns. And the brain is very large, and also it's somewhat mobile. So as an animal is moving around, the brain moves very slightly. So it becomes very difficult to record a single neuron from a moving animal. And nobody really knew how to do that for a long time. But in the 1960s, some techniques came along, and in particular, a technique of using flexible microwires was developed. And one of the people who was really instrumental in making this happen was Jim Ronk in the US. And he started recording from hippocampus using these flexible microwires. And he had found that neurons in the hippocampus were very multimodal. So they seem to respond to lots of types of sensory information. So about this time, John O'Keefe came to UCL. So he was a young scientist who was starting his first permanent position at UCL. And he decided to use this new flexible microwire technology to address this emerging question about the hippocampus and did it have something to do with memory. So he began recording from animals as they were moving around freely. And this is the paradigm that he started using, which we still use today. So you can see that in the schematic there's an animal walking around an open arena, so a square platform, and it's being monitored by an overhead camera, and there are electrodes implanted in the hippocampus that are detecting action potentials. So you can see the oscilloscope trace on the right is showing the typical burst of spikes produced by a single neuron in the hippocampus. This is what we call a complex spike burst. It's very typical of hippocampal neurons. And if you note where each of these action potentials took place in the environment, so where was the rat when that cell produced that action potential, you can see on the plot on the left the little squares are all kind of accumulating in the same place. So that was O'Keefe's discovery, that if you monitor the action potentials from a given hippocampal neuron, they will tend to be produced in a particular region of the environment, which we call the cell's place field. So here's an example of a sort of a more dynamic representation. So we're showing here two cells, one whose action potentials are in red and the other whose action potentials are in green, and they have place fields in different parts of the environment. And you can see that the rat's moving all around the environment and moving between these two locations. But each time it returns to one of those locations, that particular cell starts firing again. So the cells are somehow tracking where the animal is so that they know when it's their turn to fire. And when we aggregate all of the spikes from a particular cell in a particular region, we call that the cell's place field. Typically, when you read papers about place fields, you'll see a place field represented as a heat plot, this kind of rainbow plot with hot colours representing firing intensity, so spikes per second. That's just a way of kind of smoothing and averaging the data so you can get a sort of an overall picture of what that cell is doing. So this is an actual recording from a rat that's walking around, and you can see the tether, which is carrying the the signals to the recording equipment, and you can hear little blips, which are the action potentials. And you'll notice that they become very intense when the animal is in the north of the box, and then when it walks away again, it goes quiet. So that northern region of the box is the cell's place field, and you can see the corresponding oscilloscope trace on the right there. So based on this discovery that cells in the hippocampus emit their action potentials in spatially localized regions, he and his colleague Lynn Nadell proposed that the hippocampus might be a cognitive map, so the long proposed cognitive map that may be the site of an internal representation of space and the environment. So they wrote this book called The Hippocampus as a Cognitive Map, which was inspired by the discovery of place cells, but which also wove together lots of other research that was emerging at the time. So they proposed that the role of the hippocampus in memory is to create a spatial map, which is used to organize memories. And we'll be coming back to that notion later on, but it's been very, very influential. So place cells provide a window into the rat's thinking about where it is and its learning and formation of new memories about where it is. And so they also provide a window into studies of memory consolidation. So they've been incredibly important for advancing our notions of how the hippocampus and the neocortex might be interacting.